Welcome. You're going to be seeing the first of a series of videos that take the lab exercises from in-lab to your home. And so it's going to call for some creativity on your part, some adaptability, but I'll show you all the things that you need to know and want to understand moving forward. So this is lab 1. It's meiosis modeling and meiosis in rye anthers. And you have the lab, you've read it over. Here's how we're going to modify things. In the lab, you'd be working with plasticine, rolling it out to specific lengths. As you can see down here, you'd be cutting this to create the image of the sister chromatids, genetically identical to each other. And you'd be placing these little beads at specific locations to represent the identical alleles on the sister chromatids. So if you have something like plasticine, Play-Doh, clay, whatever, you can exactly mimic what's happening here. But if you don't, get creative. Maybe you have some little magnets that you could devise to represent the identical sisters and how this homologous pair synapses with another homolog. Whatever way you come up with, I'd like you to create the various stage images that are shown in lab 1. In addition to the sort of Play-Doh version, and I want to stress that while this might be sort of fun and interesting, and if you have kids, get them involved as well, but this understanding of how meiosis, the chromatids, the genes and alleles behave is so crucial to understanding the Mendelian portion of genetics, which is really all those things where we take individual chickens, theoretically, and cross them and look at the numbers and kinds of offsprings, the phenotypes and genotypes. So it is really important to have this as a very powerful background to then move on to understand how genes are transmitted from one generation to the next. We'll also be showing you some specimens. Now, here are my rye heads. I grew these on the farm at Salt Spring, and later on I'm going to be dissecting these, taking out the male germ tissue, the pollen anthers, crushing those and seeing if we can identify different stages in the meiotic process. In addition, I'll be showing you imagery of prepared slides from Lily in order to make sure that you see every different stage that you're meant to do.